As you can see from the title, the topic today I'm going to give you guys are an exciting kind of study that is focusing on the lineage plasticity events in lung adenocarcinoma, particularly in the early stage lung adenocarcinoma, by using the single cell based multi-omics technologies. So let's start. Oops. Okay, so at the very beginning, I want to give you guys a brief background of this study. As we know that lung cancer was still the leading cause of cancer deaths, and with the gradual application and the kind of availability of the low dose CT screening method, more and more patient was diagnosed at the relatively early stage. And this situation proposed a kind of urgent need for us to gain a deeper understanding of what's going on within the early stage lung adenocarcinoma patient. So that's the brief background of this study. And then I want to give another brief introduction of what we have done previously, focusing on the lung cancer project. So here on this slide, it showed a brief introduction of the unique kind of sample design for the study we have performed on lung cancer. So for every patient, like in our study, we have collected four tissue areas. So here, including the tumor area, the three kind of normal lung area that are coming from the same patient. And using this kind of design, we are able to gain a further understanding of the changes related with this concept because it's field cancerization. And this is one of the key and most important concepts that are kind of gradually developed during the recent decade regarding the lung cancer studies. And this one was already published in 2021. We are able to show a brief kind of atlas of the single cell level landscapes within the early stage lung adenocarcinoma. And then on this slide, I show a little bit further details about the previous two studies from the same cohort that are focusing on the findings we have made. The first, we are able to depict the tumor microenvironment changes. I mean, regarding the abundance changes for various, immune cells and stroma cells. And what we found here is interesting kind of enrichment in the B cell component in the tumor samples compared with those normal regions. And more interestingly, we found the enrichment of B cells was kind of more specifically, enriching the smokers compared with the non-smokers. And we were also able to characterize the B cell clonalities, along this multiple samples that I have showed on the previous slides. Okay, so this is a brief introduction about the background. And then now I'm going to show the design of the study I'm going to tell you today. So in this cohort, we enrolled 16 patients, and for each patient, we did similar sample collections. That means for every patient, we have collected 4 samples, including the tumor and the adjacent normal lungs. And after we got the samples, we profiled their single cell transcriptomics, as well as the whole exome sequencing to get an idea of what's going on within the tumor cells and normal cells. And with this data, we perform integrative bioinformatics analysis, trying to build a comprehensive, pictures and atlas of the single cell we identified, as well as for example, the tumor cell meta programs that we can get from this data. The final goal of this study was there are 2 points. First, we want to understand the tumor cell characteristics, especially the tumor cell heterogeneity that are involved in this early stage lung adenocarcinomas. And second one, we want to hunting for the populations that have that shows lineage plasticity events in this cohort, and trying to establish a connection between the tumor cells and the normal cells that potentially give rise to this tumor cells. This is kind of trying to answer a cell of origin problem for this lung adenocarcinoma cohorts. All right, so the first part of this study is on this slide providing a landscape or atlas of the single cell populations we have identified. In total, we have captured around 230,000 epithelial cells. And by a further subclustering, we are able to divide them into airway cells and alveolar cells. So majority, as you can see based on this slide, the majority of the epithelial cells was alveolar cells. And within the alveolar cells, we have 4 subpopulations, which are shown here using the numbers. We have alveolar type 1 cells. We have alveolar type 2 cells, the most 2 abundant cell types. And in addition to these 2 major cell types, we also identified 2 populations which show kind of intermediate features, which we call them alveolar intermediate cells and bronchial alveolar cells. So both of these 2 population, they show the kind of a combination pattern regarding the markers of alveolar cell lineage specific features, as I show on this bubble plot. In addition to these alveolar cell populations, we also identified several airway cell populations. Because of the large number of cells captured, we are able to identify several rare cell populations here, including, for example, ionocyte and tuft cells. All right, after we got the overall idea of what we have in the normal epithelial compartment, we start to using comprehensive computational tools to determine what's the features of malignant cells. So by using the inferred copper-number origin features as well as the expression profile clustering features, we are able to identify around 17,000 malignant cells from all the tumor samples of all this patient. And on this slide, it shows a clustering plot that are colored by either patient and the copper-number ordination scores. So what I want to point out here is that you look at the copper-number ordination scores, and you will see the copper-number ordination for the malignant cells shows a high level of intertumoral heterogeneity, illustrated by the distinct color patterns of this middle UMAP. And then if we look at the per sample cluster, you'll find a clear gradient pattern coming from right to the left, indicating the copper-number ordination level show also an intratumoral heterogeneity. And those malignant cells, if we look at the gene expression profile for the lineage marker genes that are selected, it'll show us a gradual decrease. I mean, the expression level of marker genes was decreased compared with the corresponding fully differentiated lineages. And then, because I just introduced for this cohort, we perform whole exome sequencing for the tumor samples as well as matched normal samples. We are able to determine the driver mutation for every patient in this cohort. And here I show, on this slide, I show a brief introduction of the driver mutations or frequent driver mutations or mutation events we identified. The most frequent mutation was EGFR, and also we found TP53 mutation as well as KRAS mutation. In addition to these 2 mutations, we also find, for example, LKB1 mutation, and we also found fusion events in one of the patients. But due to the the limit cohort, we focusing on the EGFR and KRAS, those frequent mutations. The first thing we did is trying to say whether these mutations can be represented or reflected on the transcriptomic level. So we perform PCA dimension reduction based on the expression profiles. And then we are able to project the mutation information on this PCA plot. So the most kind of striking difference we observed is the malignant cells coming from the KRAS mutant sample seems to be quite different from the remaining malignant cell that are coming from the non-KRAS mutation samples. So the KRAS the malignant cells are distributed along this PC1, which indicate like those cells are, I mean, the variation within this data cohort shows the largest variation on this like KRAS mutation signal. And we further quantify this difference by using Bankchaya distance by comparing the malignant cell distance between different driver mutation groups. You will see here like the KRAS, the distance within the KRAS samples are lowest. And if we calculate the distance between KRAS mutant samples and KRAS wild-type samples, the distance was increased dramatically. After observe this phenomenon, we further quantify additional features of the malignant cell. So one of the features we could get is based on this published study. This beautiful study coming from the Stanford University indicate like at single cell level, people can use the transcriptomic complexity as a measure to predict the cell differentiation level or cell differentiation status. So based on this idea, we ran a computational tool that is called Cytotris to predict the cell level differentiation status. So this slide shows the prediction result. On the very left slide, I show the same part of the malignant cells on a UMAP that are, trying to show the difference at mutation group level. For example, the KRAS mutant one on the top part and the wild-type one on the other part, and particularly you will see like EGFR mutant one on the bottom right part. So with this UMAP, after I project the Cytotris score, which is a differentiation score on it, we found the KRAS mutant malignant cell in general, they are less differentiated compared with other mutation groups. And then when we look at the copper number ordination profiles, we also found the malignant cells coming from the KRAS mutant group. In general, they have a lower, copper number ordination profiles compared with the EGFR ones or the other patients. And then finally, for the malignant cells, we want to quantify them at a relatively high level, at a metaprogram level. So currently, because the research field are accumulating more and more single cell data, and with this large amount of data, we're able to quantify or categorize the general high-level metaprograms at a relatively kind of robust level. And then this slide shows two individual studies that are published. They try the central goal of those two studies is trying to give us a consistent or relatively consistent metaprograms that are summarized or like synthesized using a computational method from multiple tumor types. So we use this wonderful like resources using their metaprogram features and trying to quantify the metaprogram levels in this lung adenocarcinoma cohort. And this slide shows a result for this metaprogram computational quantification result. On the left part, the heat map shows the unsupervised clustering of the metaprogram. So here, each row corresponds to one metaprogram, and each column corresponds to one malignant cells. With this unsupervised clustering, you will see like the malignant cells coming from KRAS mutant samples. They show quite different patterns compared with the wild-type ones. For example, those KRAS mutant malignant cells are enriched in this cluster 1, which is colored by this red colors of this driver mutation label. And this C1 cluster, they are enriched for signals, for example, metaprogram 19, which stands for epithelial synthesis, and metaprogram 13, which stands for the KRAS signature that are compiled from multiple cancer types. So this further gain us more confidence, like trying to pursue further on this KRAS mutant kind of malignant cells, because they show quite different transcriptomic features, whether it's on gene level or metaprogram level. And so as one part summary regarding the malignant cells in this cohort, we're able to show like their malignant cells show higher level of tumor heterogeneity, and then the malignant cells coming from the KRAS mutant sample, they are different. They show quite unique transcriptional reprogram patterns. So the next question we asked is whether we can find connectors or bridge or kind of progenitor cells that are from the normal cells part from this epithelial cohort that can link the malignant cells, especially the malignant cells coming from KRAS mutant lower samples. With this question, we went back and looked at our data. So the first point that caught our eyes is there are two populations from the alveolar part that shows, intermediate expression states regarding the lineage plasticity genes, lineage specificity genes, I'm sorry. They are the alveolar intermediate cells as well as the SDP. I'm sorry for the name confusion, so I forgot to rename it. So this one stands for the bronchial alveolar population, which I mentioned previously. So these two populations show kind of intermediate state. And if we look at the UMAP clustering result, I mean, the position of those two populations are in the kind of bridge part that are connecting the 82 cells with 81 cells. So we think those two populations are of potential interest that we can pursue further. That's what we did. We checked the fraction changes of those cell populations. The basic idea is like, if those cells are related with tumor genesis, then probably they have kind of enrichment or depletion patterns if we compare the tumor samples with the normal samples. That's pretty straightforward idea. And by checking the fraction data, we found the AIC cells, alveolar intermediate cells, show this interesting pattern. They are significantly enriched in the tumor samples compared with those normal regions. And this SDP, they didn't show significant result. And by looking at the per sample level fraction result, we confirm the alveolar intermediate cells has an interesting pattern of enrichment in the tumor samples. So we decided to go further on these AICs. So we did two analyses. First, we constructed the developmental trajectory by merging the AIC, by grouping AIC cells, alveolar cells together. We want to construct a potential developmental trajectory path and see what are the AICs functional as. So here on this left part of the slide, I show you those AIC cells are located at the intermediate states, which correlate with the previous findings, like showing they are the potential intermediate cell that bridging 82 and 81 cells. And then the most interesting part is like this trajectory. I mean, coming from stemming from 82 cell, they have two branches. On the lower branch are the normal 81 cell branch, and on the upper one we find our malignant cells, and particularly at the very terminal or the tail part, we saw the enrichment of the malignant cells that carrying KRAS G12D mutation. This is a very exciting result. And we decided to further look at those AICs and try to see what are those cells that are giving rise to malignant cells. And the thing we did is trying to say whether those, because here you see the AIC cells was distributed on both branches. We think maybe they are heterogeneous. And by checking the distribution of the cell differentiation level, we are able to find two peaks. There's the peak which are enriched in lower samples, and another peak which are relatively depleted in lower samples. And this CAC cell that are enriched in lower sample, they show less differentiation score, which makes sense, right? Because those cells are supposed to like more kind of de-differentiated if they are the progenitors. And then we did subclustering. We pull out those AIC cells, perform further clustering, and we are able to find two clusters. On the upper part, we have a orange part, which is which has a high expression of marker genes like KRT8, CDKN2A, CDKN1, and Claudin4. And on the lower part, we have the remaining cells that we call them as other AICs. The other part, we call them as CACs, like KRT8 positive alveolar intermediate cells. So those CAC cells, if we look at the enrichment level by going back to the fraction data, and we found those CAC cells fraction are more significantly enriched in lower samples compared with the AIC cells enrichment result. This gives us further confidence about this findings. And then we perform further kind of validation analysis and discovery analysis using the independent technology platform. The first thing we did is we quantified or profiled the tumor microenvironment using a technology called digital spatial profiling, DSP, on one of the patient 14, for the tumor red areas and tumor adjacent normal areas. And the interesting findings from this DSP analysis is not only we are able to validate the marker gene expression in the tumor area, but also we found that those markers of CAC cells was relatively kind of enriched in the tumor adjacent areas, but not in the involved normal lungs. So this is very interesting because it gives us more clues. Like if we consider the features of proximity of those marker genes, and also it indicates us the potential involvement of those CAC cells in the early stage LUADs. We also perform 10x spatial profilings using the same patient. This is also patient 14. And we are able to histologically annotate the major cell populations. On this slide showing the spatial profiling of patient 14, we are able to identify two tumor areas, which are color labeled by brown colors. And then in the middle part, we have the reactive pneumocytes. For the reactive pneumocyte, we are also be able to dichotomize them into two groups. We have the more kind of reactive group and less reactive group. The more reactive group are serving as again a bridge that are connecting those two tumor regions. So this spatial profiling provides a very interesting and important validation, and spatially it gives us more kind of confidence, like showing the CAC's involvement in the early stage lung adenocarcinoma malignant cell development. And then we perform computational validation using the public cohort. So by separating the CAC cell from the normal cells, we are able to determine a CAC cell signature using the genes that we identify as marker genes or differentially expressed genes. Here it shows the CAC signature levels by comparing the lung adenocarcinoma samples versus the whether it's normal samples or pre-kind of malignancy stage samples labeled as AAH in this pre-malignant cohort. For both cohorts, we identify this CAC signature was increased, especially in this malignant cohort. This CAC signature was showing increasing gradient trend. This is very interesting, and then we are able to validate the prognostic power of this CAC signature. Those samples, from the TCG lung adenocarcinoma cohort, the samples with low CAC signature, in general, their survival was better than the high signature group. From this different aspect, we are able to validate the importance of the CAC cells involvement in lung adenocarcinoma early stage development. And finally, if we map those CAC cells back to the trajectory we have predicted, we saw a clear trend, like those CAC cells compared with other AIC cells. This inlet panel shows the cell that are identified as AIC. And then the brown colored ones are those subset that are identified as CAC cells. You will say those CAC cells are more enriched in the malignant cell development branch. All right, so at this point, we identify CAC cells, which are serving as the potential connectors or the progenitors of lung adenocarcinoma development. And we want to validate them in a more kind of systematical way. So, okay, so this slide show additional evidence for like potential involvement of CAC cells and KRAS mutant features. So we cannot compare the CAC cell score or CAC cell signature score between KRAS mutant malignant cells and KRAS wild-type malignant cell. Here, the EM-LU80 means EGFR mutant ones, because this one has the most number of sample size we can use. You will see like the malignant cells coming from KRAS mutant ones are showing increased CAC signature, indicating the strong correlation between CAC cells and KRAS mutant lung adenocarcinoma. Considering this, we used a very interesting genetically engineered mouse model, which is a KRAS mutant mouse model. With this mouse model, after the treatment, the tobacco carcinogen simulation treatment, the mouse cells, the mouse can develop, can grow the tumor cells within the lung tissues, and we use this model to validate the involvement of the key findings in this human part. What we did is, with those mouse model, we captured the cell. We collected the cells at baseline, which is end of the tobacco carcinogen exposure, and 7 months after the exposure. And using this 2 time point, we want to gain overall kind of cellular abundancy dynamics regarding tumor cells and the CAC progenitor cells. So we perform single cell RNA sequencing for those 2 time point samples. Within the epithelial cell, we identified equivalent cell populations in the mouse model. We identified, for example, 81 cells, 82 cells, as well as CAC cells. So the CAC cells are also located in a very similar location on this UMAP. They are serving as a junction or bridge between the normal alveolar cell populations. And we checked the tumor cell fraction as well as the CAC cells from mouse data. The tumor cells was showing up in 7 months after exposure, and the CAC cells shows the abundance change at the early time point at the end of treatment. And then at 7 months time point, it shows kind of a difference, but this difference is not significant. It has a high variation. And this indicated the early involvement of these CAC cells in the mouse model. So in addition to that, we did similar analysis using independent technology platforms. Here we use 10x spatial profiling platforms to profile the lung tissue that we collected from the mouse model, trying to see if we can capture interesting phenomenon that we observed in human samples. So this one shows one of the sample we have included in this study. So in this sample, we observed 2 kind of tumor areas, which is colored by this brown dot, and then surrounding these 2 tumor areas, we found a lot of reactive pneumocyte dots colored by the purple dots. And then if we after we quantify the CAC signature using the mouse data, we found the CAC signature. They show an interesting gradient. If we look at this signature score colored by the red band, you would say like they are. The signature score was highest in tumor body, and then in the surrounding areas, which is enriched for reactive pneumocyte, the CAC signature score, the CAC signature show up. And again, this CAC signature was strongly associated with KRAS signature. So with this mouse data, we are able to computationally predict the trajectory developmental paths. We pull out the alveolar cells as well as the CAC cells and constructed a monocle trajectory analysis. From these slides, you will see like those cells, again, they are stemming from the alveolar type 2 cells. And along this trajectory, we observed either tumor cells or alveolar type 1 cells. And in the middle of the trajectory path, we observed our alveolar intermediate cells. So here, aka the CAC cells. And by taking a deeper look at the pseudotime as well, pseudotime means the the matrix that we use to that we derive from the trajectory analysis. It shows us a predicted time order regarding the cell development. So if we look at the pseudotime data, it shows like those CAC cells are like serving as an intermediate part between the alveolar type 2 cells and the malignant cells. And those CAC cells are showing, the cytotrace score indicating the cell differentiation level show the same kind of distribution patterns. And in addition to that, we are able to screen for the KRAS mutation at single cell level. This one served as a independent validation for the involvement of CAC cells that are coming from the mouse data. From those mouse CAC cells, we observed a dramatic fraction of cells that are carrying KRAS G12D mutation. This again gave us more kind of clues like those KRAS, those CAC cells are involved in the mouse models lung adenocarcinoma development. In addition to this experiment, we are during the revision process, we are requested to perform a more kind of straightforward analysis to show that those CAC cells are involved in the lung adenocarcinoma development. So in general, currently there are two models. If we consider the cell of origin problem for lung adenocarcinoma, the majority of one of the group, one of the research community, they believe like in general, 82 cells are serving as the cell of origin for the lung adenocarcinoma development, because like 82 cells, they based on kind of independent stemness or cell reprogramming analysis, or those 82 cells, they show kind of higher stemness features compared with the other alveolar cell components. So here we adopted the same hypothesis. We hypothesis are like in our data set, the actual cell development order was coming from 82 cells and then goes through this alveolar intermediate cells and goes to either 81 cells or tumor cells. And then this mouse model experiment shows us that those CAC cells are actually derived from the 82 cells. So we use the same mouse model, but this mouse, when we collected for the samples for single cell RNA-seq, we try to label the 82 cells specifically using the SFTBC genes as a marker. So we collected the cells that are 82 positive, and then we perform single cell profiling for those 82 labeling cells. And in the middle, we show the single cell clustering result of those cells that are coming from 82. You will see like those cells that undergo the 82 cell states. They show diverse of cell population feature. Here we captured those 81 cells as well as intermediate cell states and the tumor-like states. And on the right, we show a consistent kind of abundancy changes by comparing different treatment groups. So finally, as ultimate validation, we adopted another mouse model to justify the malignant cells we observed are actually coming from A2 and why this goes through this CAC cell states and from tumor cell. What we did here, we use another mouse model, which is able to labeling KRT8 CAC cells that we chose as the marker, the KRT8. And we performed additional image profiling. Here, we are comparing the tissues we collected from the mouse at different time point. We have one time point right after treatment, and then we have multiple time point after the treatment. You will see here, if we compare the tissue areas coming from normal appealing images and the tumor images, there's a strong kind of signal captured by either TD tomato or KRT8, indicating those cells are the KRT8 positive and cells that are undergoing the CAC cell states. We also compare the fraction of TD tomato positive tissues, and we found a significant increase for the TD tomato positive tissue areas, comparing follow-up with the end of treatment samples. Because the KRT8 was not specific to alveolar cells. So here we use LAMP3 as an additional marker to ensure that the tissue areas we captured are actually the CAC cells coming from the 82 cells. By comparing this TD tomato positive, LAMP3 positive cells fraction, we observed a strong increase in the this subtype of cell population in tumor samples. And this justified our hypothesis, like those tumor cells are coming from 82, goes under the CAC cell state and form the tumor cells. So as a summary, the take-home messages. First, this study established kind of a modularized computational network, trying to combine different computational analysis modules to comprehensively discover the progenitor cells, to validate the potential involvement of those progenitor cells during the malignant cell development. And second, we are able to justify the malignant cells shows the loss of lineage specificity by looking at their gene expression markers, the marker genes expression, I'm sorry. And then thirdly, the KRAS mutant lung adenocarcinoma showed a less differentiation status and very consistent transcriptomic reprogramming patterns regarding the meta programs we included in this analysis. And finally, our trajectory analysis, as well as the computational analysis from the genetic mouse models, suggest the malignant cells coming from KRAS mutant lung adenocarcinoma developed through the 82 derived AIC cells, here specifically the KRT8 positive alveolar intermediate cells. So finally, I want to make an acknowledgement for the research group, the people that are involved in this study, as well as grant support for this to make this this study coming true. And also, I want to give thanks to the patient and their families for their key and important and key contribution to this study. So I guess I will stop here and take any further questions if you guys have.
Well, thank you very much, Dr. Han. That was a wonderful presentation and a lot of exciting data. Maybe you could, great. I was going to say we might have questions for the slides. So at this time, if anyone has a question, you could feel free to put that in the Q&A. You can also raise your hand and I will call on you and let you ask your question. So maybe I can get us started. Again, really interesting, and I have many questions for you since we're very interested in this topic as well. I was curious, you showed in the very beginning that in your tumor samples that you could identify cells that looked like AT1 cells, AT2, the alveolar intermediate cells, and you also had a cluster that was like bronchioalveolar cells.
Yep.
And of course, this is very interesting to me because I think you listed that they express SPC and a secretoglobulin. So they're very similar to a cell that we study in the mouse called BASCs. I was
Bags.
curious if you have studied that population any further. Is that population also linked to the tumorigenesis, or what can you say about them?
Yeah. First, thank you, Dr. Kim, for your this is for interesting and key question. I think this is one of the key questions that I got the same question, similar questions, not from only you, but also additional scholars. But unfortunately, currently we didn't push further on this direction, like from this cohort, like whether this BAX, whether it's you call it BAX or the bronchial alveolar or the SDP, so this population, whether it's further involved in the tumor cell genesis. So I think, nonetheless, I mean, it's still important because those cells are another major, during my presentation, I didn't mention that. In addition to the alveolar intermediate cells, those cells are one of the important population that when we consider lineage plasticity, right? Because they serve as another kind of hypothesis or potential of cell origin that can explain the tumor cell genesis. In the current study, we didn't push further on this, but to my understanding, we can do more. Definitely, we can do more on this. First, because this one, based on the current study, the fraction of those back cells, I mean, based on your description, they didn't show any enrichment in the tumor samples. But we have multiple explanations for this. If we look at the sample level distribution, we saw those back cells are particularly coming from one patient, the patient 13. So they may be potentially involved in the tumor genesis, but because of this kind of data bias, we are not able to capture the association. That's one of the possibilities. Considering this, I think we can do more on the potential involvement of this SDP. We can try to screen for the KRAS mutation or other involved mutation that we can do for this cell population, or we can further, infer computationally quantify the trajectory development of process for this population. Yeah, I think this will be a very exciting direction that we can explore further. Thank you.
Wonderful, thank you. Any questions from the audience?
Maybe I have one question. Guangchun, I think you did the trajectory analysis by using slingshot and also molecule 2,
Oh, yeah.
I think.
Molecule 2 and slingshot, yeah.
Yeah, and several similar software, since we actually did bioinformatics analysis here in the team. So I was just wondering, could you share any experiences you have by using those 2 different software, and why you actually try to use 2 different software? Just because get different result, or any preferences you have?
Oh yeah,
that would be great.
Yeah, so at the very beginning of this analysis, I mean, at the first submission for the trajectory prediction, we used monocle 2 and cytotries, because cytotries are one of the quantification method for the de-differentiation level. It helps us to determine the routes for the development order. So we use this too. And during our revision process, we are required by the other additional reviewers, to provide the robustness of our analysis. So based on that, we tried 3 additional trajectory construction method. And after we tried them, we are required to show the consistency level. And based on that result was not shown here, but we are able to observe a quite high consistency regarding the differentiation levels between, like, for example, monocore, solid trees, and slingshot, as well as there's another two softwares which I couldn't indicate the name now. But yeah, we in total we tried 5 trajectory construction method as well, in addition to the saddle trees analysis. So to make them, kind of robust.
I see. Thank you.
Thank you.
Other questions?
Yeah, we
So
have one question from Ashish, I
Yeah,
think.
so
Right.
I do have one question. Thank you, Dr. Han, for this really good presentation. So like I do have one question on the spatial transcriptomics. So basically, which basically tools you use for the annotations of the spatial data?
Thank you, Dr. Jan, right? Did I pronounce it right?
Yes, thank you.
Yeah, gotcha. So are you talking about this spatial data? This one, like the result?
Yes, yeah.
Okay, I mean the histological annotation part was done by my colleagues, like the histopathologist. So in the very beginning, I think they use the dedicated tools. I mean, regarding the image analysis, like quantification of cell level expression. But after that, when we do the spot level annotation, because at the very beginning, we can only do the region level annotation. And after that, based on the expression, we can do the spot level one. Regarding the spot level one, we use the loop browser that are published by 10x to achieve this goal. Did I answer your question?
Yeah, sure. Thank you. Yeah.
Thank you.
This is Carla. I was wondering if it is possible for you to overlay the data from the human samples and your mouse samples to see how similar are the CACs in the mouse to the human CACs? And is that a useful type of analysis to then identify targets of interest?
Oh, I got you. This is a perfect question. So this is also one of the reverse questions regarding the robustness of what we would say the consistency regarding the marker genes, if we consider the therapeutic potential, right, that we could identify. So we performed overlap analysis regarding the markers from human and the markers from mouse. We took the overlap and we quantified the proportion of overlap part within each compartment. The thing we observed is like if we try different cutoffs, we take the top 20, top 50, top 100, top 300. The overall trend is like when we include the most top ranked genes, we observed gradually increased kind of enrichment. That's one of the signs for the conservative regarding the marker genes. And second point, it's like for the important genes we show in the result, like the bubble plot or the marker genes in the additional kind of data figures. Those genes were observed in both human and mouse. So suggesting, yeah, they are very conservative.
Great. And I think when you focused on the CACs, you are focusing on the KRAS mutant samples.
Yep.
But maybe I misunderstood. I was curious if the CACs in KRAS mutant cancer are similar to those in the EGFR mutant, or you don't see them in. I know you showed there were more in the KRAS mutant, but when you find the CACs, are they similar in the different genotypes?
This is a wonderful question, and this is one of my I made one potential kind of future research direction for this. Anyway, yeah, this stands for a very interesting question. We will consider lung adenocarcinoma based on the different driver mutation background, right? EGFR stand for one of the very popular subtype, and usually they show different levels of differentiation level or carbon number alteration levels. We compare them with KRAS. So we do observe CAC cells in EGFR mutant lower samples. And second, as I show you, and you also mentioned, the CAC signature score was kind of decreased when we compare the EGFR ones compared with the KRAS mutant ones. So this is my temporal thoughts regarding this. I don't have a data support for this, but here's my my comments, my 2 cents. I'm sorry. So the CAC signature was significantly associated with KRAS mutant malignant cells in this cohort. This is one of the findings. And regarding the EGFR ones, I think if we consider the developmental trajectory of malignant cells with regard to the mutation status, I think KRAS mutation and EGFR mutation are different. So maybe KRAS mutated malignant cells are showing less differentiation, or vice versa. So that will that will add more kind of complexity, making this CAC signature analysis kind of challenging. So that's what we are doing now. We want to capture more malignant cells coming from both groups. I'm sorry, not malignant cell, the CAC cells. We want to capture more by accumulating more sample size. And then we also want to investigate the cellular level features at different layers. For example, the epigenetic layer. We want to know the regulatory events that are involved in the CAC cells regarding this mutation background, because we think this is involved in this question. So yeah, this will definitely be a very interesting research direction that we can pursue further. I'm very glad for these questions. Cool.
Wonderful.
Thank you.
Okay. Any other questions?
Hi, Dr. Han. Yeah, this is great talk. I wonder, because you mentioned about the KRAS mutant and EGFR mutant, what about the MET mutant?
Okay, for the MET mutant samples, we do compare them, but the major issue here is we only have one here. The MET mutation we only have one patient, and this patient is patient 4, and it is not we didn't capture so much cells when we consider the cell number, so we didn't get a reliable statistical result. So that's why we didn't put very strong kind of statement on that. But for example, we do compare their malignant cell transcriptome features. For example, here in this Barchaya distance analysis, which is trying to characterize the transcriptomic heterogeneity or diversity of malignant cells, we saw this EM, I'm sorry, not EM, the MM, which means MET mutation. The MM patient was showing kind of relatively comparable levels regarding the transcriptomic complexity compared with the EGFR ones, or the other like ERK fusion ones. So we know MET mutation or ERK fusion are sometimes they stand for a very aggressive phenotype. So we think, I mean, with more and more accumulating patient cohort, we are able to answer your question at a more comprehensive level. But this is a really important question. Thank you.
Thank you.
Yeah.
Great. Anyone else? I think we're almost at the hour, so we just want to thank you, Guangchun, for a really interesting discussion. and thank you for this invitation. It was great to be part of this.
Thank you. Thank you, Dr. Kim and Dr. Liang for inviting me. And I really appreciate discussion with you and trying to show like what we have done here. And we wish more collaborations with you guys in the near future. And by the way, I want to make a small advertisement. I'm trying to become independent, trying to find a position. If you guys have any, came across any ad or whatever, just let me know. Thank you.
All right, thank you so much. Well, thank you both. I think it's a great talk.